Do you want it's okay. Okay. So first, I would like to thank the organizing committee for inviting me to make this presentation on genomic of arthrogryposis. And so just to, I mean, maybe there is some redundancy with the previous talk, but so just to remind the definition of AMC, which is a developmental condition characterized by joint contractures in two or more body areas, and it's a direct consequence of reduced fetal movement. And this reduced fetal movement is responsible for muscle atrophy, for pterygia, as you can see here, macrognathia, pulmonary hypoplasia, and diaphragmatic defect. So it's not specific, but it's rather it's a consequence of reduced fetal mobility. The incidence is estimated to be 1 in 3,000 to 5,000 live births. So, as mentioned by Joanna, there are multiple causes of AMC. There are first intrinsic causes leading to limitation of fetal movement, such as multiple pregnancy and oligohydramnios, maternal immune disease, such as myasthenia gravis, congenital infection with CMV, varicella, rubella, and more recently Zika. Chromosome abnormality, I think this is very important to remind that we have to perform RSCGH during pregnancy and after, if needed, because these chromosomal abnormalities are responsible for 6% of syndromic AMC, and there are multiple causes of genetic causes of AMC that I will summarize now. A total of 402 genes have been reported so far in AMC. So the aim of our large series of the investigation that we perform is to improve the genetic diagnosis rates of AMC, as well as to identify new disease genes. So in terms of patient inclusion, we include a total of 315 unrelated families, including either non-syndromic AMC, which represent 206 patients, or syndromic, and you can see how frequent is this syndromic, although there is a bias because in this case the diagnosis was not established, of course. So this is thanks to the collaboration of pediatrician, neuropediatrician, fetopathologists, and clinical geneticists. So the sporadic cases were observed in 226 families. 45 of them were born from consanguineous parents, 151 from non-consanguineous parents, and it was unknown in 30 families. Familial cases were observed with at least two affected families were observed in 89 families. And parents were consanguineous in 30 families and non-consanguineous in 54, unknown in 5. So the AMC diagnosis, I mean in terms of clinic, was established during pregnancy in 251 patients out of 3,315 families, so 80%, through ultrasound examination. And it was established at the first trimester in 86 families, so 34%. During second trimester ultrasound examination in 103 families, so 41%. Third trimester in 27 families, not reported in 35 prenatal cases. The diagnosis was established after birth in 62 families, so in 20% of families, and it was unknown in 2 families. So voilà. So the AMC was classified as non-syndromic versus syndromic, depending on the associated feature, unrelated to arthrogryposis or unrelated to reduced fetal movement. And in 109 family, the AMC was syndromic, so in 34% of cases. And here, you can see that the most frequent associated features were intrauterine growth retardation in more than 9% of cases, epilepsy, intellectual disability, and brain malformation, so in 6 to 11%, congenital heart defect in 4%, and hemangioma in 8%. Of course, this syndromic form may be diagnosed before, but also after birth, especially for hemangioma and for epilepsy and so on. In 25%, in 25 cases, the AMC was diagnosed during pregnancy, but not the associated features, which is a critical point for the information of parents during pregnancy. Okay. In syndromic AMC, both AMC and additional clinical features were discovered during pregnancy, so 66% of cases. In 22 affected individuals, so 20% of patients, both AMC and associated feature were discovered only after birth. And so, as mentioned, in 23% of cases, the AMC was diagnosed during pregnancy, but not the associated feature, including brain involvement in 17 cases. And this is a critical point, I think, about the problem of syndromic AMC. So the genomic strategies that we apply for in the frame of research, I have to mention, is first, it was genetic mapping a couple of years ago when the whole exome was not yet available. And so it was performed in familial cases and or consanguineous families. And the gene was identified in 100% of cases. Targeted exome sequencing, as you can see, only 32% of genetic diagnosis was established at that time. But as you can see, as you will see, it's mainly because of new genes, which represent a high percentage of our cohort. And so we moved to whole exome sequencing, and the gene identification was established in 61% of cases. I have to also to mention that when, for example, in this case, there was no diagnosis, no gene identified, it's important to remind that we have to perform CGHRA, SMN1 gene analysis, because it could be associated with arthrogryposis, and also DMPK for myotonic dystrophy, which is also associated. So this is important during pregnancy to propose this associated examination. So among the genes that we identify during this the last couple of years, you can see the frequency of gene identified in this cohort of patients, and you can see that in blue, these are the gene which we identified recently, we or our collaborators, in the last 6 years, and it represents 21% of a disease-causing gene, which is a higher percentage. And it includes a couple of genes that I will come back to. So in this schematic representation, you see the neuromuscular system with, you recognize the skeletal muscle, the neuromuscular junction, the axon, the cell body of motor neurons, and also the brain. And you can see that first, that in red are the genes that our team identified. And you can see first that about axoglial defect, which means the defect of axon or myelin, of peripheral myelin. There are a couple of new genes, CNT and APR, which encode CASPAR, gliomedin, ADC6, and LGI4. And importantly also, you can see that the brain involvement, which represents 22% of cases in our cohort of patients. So this is highly, I would say it's highly frequent. And so the new gene represents 21% of the genes identified in this large cohort of patients. In terms of pathogenic mechanism in AMC, so skeletal muscle represent 40% of cases, and the second cause is brain, with or without spinal cord movement, and it represents 22% of cases. And this is important because for skeletal muscle, you may have a very good and efficient rehabilitation. And when you have brain involvement, it could be different because you have intellectual disability, and so the prognosis is a little bit different. So about the new genes that we identify, I would like to mention the GLDN, which encode gliomedin, which is an essential component of the node of Ranvier, which is located here. And we identify the mutation, pathogenic mutation in 4 unrelated families. And interestingly, we identified before CNT and AP1, which encode CASPAR, which is also a component of the node of Ranvier. And you can see the link between gliomedin here and CASPAR here. So this is a critical, there are critical protein linking the Schwann cells to axon and to maintain the node of Ranvier, which is a critical structure in the nerve to allow the potential action, the action potential to along the nerve. So we also identify loss of function in LGI4 in collaboration with Bruno Reversade in 4 families. And in this case, LGI4 is interesting, also a new mechanism because it's a ligand involved in Schwann cell myelination, and it's secreted by Schwann cells and leading to a dramatic absence of myelin, as you can see here by the fantastic work of Anila Carrière, who showed that you see in AMC, with respect to control, that you see very clearly the myelin, and here there is no myelin at all. So LGI4 is a protein expressed by Schwann cells, then and secreted by these cells, and this protein will link to ADAM22 in peripheral neuron to and it's the clear myelination signal. So that means that it could be also LGI4 could be also regarded as a novel therapeutic target to stimulate the myelination in human. And another gene also is MAGEL2. MAGEL2 is localized in chromosome 15q in the locus of Prader-Willi syndrome. And we observed in 3 unrelated families truncating mutation of this gene on the paternal allele with a feature resembling Prader-Willi syndrome. And this is quite interesting. That means that you have a large clinical spectrum with mutation of MAGEL2 from syndromic intellectual disability or autism with Schaff-Young syndrome to arthrogryposis. And finally, more recently, we identified de novo mutation in SCN1A. SCN1A, which is so responsible for arthrogryposis. And you can see these are family, 3 families in which de novo mutation were observed in 3 families. And if you look at the mutation, which is here in red, with respect to mutation in these genes which are responsible for epilepsy, there is no specific position of mutation leading to arthrogryposis. One of the 3 family develop a drug, one of the 3 patients develop a drug-resistant epileptic seizure after birth. So variants in these genes are responsible for Dravet syndrome, an early onset encephalopathic epileptic, general epilepsy with febrile seizure plus, and familial hemiplegic migraine. So our data enlarge a group of AMC linked to central nervous system involvement caused by variant of gene encoding channel like NRLCN. And this SCNA is expressed at the axonal initial segment here. This segment is critical to integrate the synaptic input and generate action potential along axon, and also to maintain neuron polarity. So that means that we have identified mutation in a gene which is also responsible for, another type of phenotype. And so that means that depending maybe on a second genetic event, maybe there is a different phenotype resulting from this mutation. So about the mode of inheritance, what we observed in this cohort is that the autosomal recessive form is the most frequent, 66%, and autosomal dominant is at the second position. Importantly, when we look at the contribution of de novo dominant variant, it's if you look at here in sporadic in consanguinous, in consanguinous and non-consanguinous, if you compare the autosomal recessive form and de novo dominant, you can see that in autosomal recessive form, the consanguinous family, of course, is the autosomal recessive form is the most frequent cause and mode of inheritance. And in contrast, For de novo dominant mutation, what we observed is that it's a non-consanguineous sporadic family. We observed 50% of de novo mutation. And this percentage is quite similar to that reported in a European ancestry from the large deciphering developmental disorder study reported by Martin et al. And so that means that even the phenotype of their patient and in this cohort, a similar and high proportion of de novo mutations, 50%, was observed, suggesting that de novo dominant mutations play a prominent part in pathogenic mechanism of developmental disease. So in conclusion, whole exome sequences is the most efficient approach with an ability to for disease gene identification in 61% of patients. In 47% of AMC cases, the genetic cause was not established. So the question is whether we have to move now to whole genome sequencing, whether there are digenic or multigenic mechanisms that we are currently investigating, or somatic mosaicism, or non-genetic cause, just to remind that before all this observation, we have to test SMN1, DMPK, to be sure that the CGHRA high resolution has been performed. So new genes identified since 2014 was found in 21% of cases, and about AMC disease gene mechanism, as mentions, the nodal Ranvier formation, the axoglial interaction for peripheral myelination are including in this new disease mechanism, but also brain development with the MAGEL2 and new developmental channelopathy with SCN1A. And the last conclusion is that the novo dominant mutation play a prominent part in a pathogenic mechanism of developmental disease. And the benefits of an accurate genetic diagnosis includes a better understanding of prognosis, and this is quite important for parents, more tailored management of AMC, and possibly other organ involvement, and improved surveillance. A precise genetic diagnosis to AMC enables an accurate genetic advice to the affected individuals and their families. So I would like to thank my main collaborators in the lab, Yéline Capri, Manuel Abusi, Flora Noland, Loïc Varek, Dana Jabair, Jérôme Baluenda, and in close collaboration with Annie Lecarrière for all the morphological investigations she performed, Marcel Tauque for zebrafish model that we perform also, Jérôme Deveau, Jean-Luc Bessereau, Bruno Versal, and Ivo Gut and Nigel Lang. And I would like also to thank the geneticists in Strasbourg, Bicêtre, Lyon, Grenoble, Pitié-Salpêtrière, and also all the pediatricians, neuropediatricians, fetopathologists, and clinical geneticists who was very active in this collaborative project. Thank you for your attention.
Yeah, thanks very much. Do we have any questions from the audience?
Hi, thank you. Very nice presentation. So I saw that in your undiagnosed cohort,
Oui.
titin is the main.
Pardon?
In your undiagnosed cohort, titin is the main gene that you find mutated. Mm-hmm. But I guess most of those cases are biallelic truncated variants, and we may, of course, have issues in the interpretation of missense variants, or we may miss other variants in titin. So what's your expectation? Would you say that may titin in general be the main key gene in the
I
field.
mean, can you repeat the main?
Yeah, I'm just wondering if
TN.
tin,
TN.
Yes.
yeah. If you think we are missing their diagnosis, and what's your expectation? Would you say that titin is probably the main contributor?
With titin, it's very difficult because it's a very large.
Can you use the microphone?
No. It's a very large gene, like ryanodine receptor, and you have to be very strictly. I mean, it should be strictly defined as what are the criteria that you will use to retain the variant. Mm-hmm.
Are there any, I find that question very interesting. Are there any additional tests that you could use for titin apart from sequencing to support?
The expression, on skeletal muscle, for example.
Mm.
I have one question.
And of course, it depends on the type of mutation, but you can also analyze the transcript. If,
And Judy, do you see any phenotypic patterns that would lead you to believe that the cause of the disease is this structural protein rather than the other proteins in the cause? And if any particular patterns?
for which disease?
Structural genes like titin, for example, or ryanodine, do they have some specific patterns that you would see that, okay, this has to be?
Oui. For example, for titin, I can come back to this table. It's, where it is, voilà, titin. So it's skeletal muscle. So that means that if you have involvement of other tissue, you can exclude. For example, nerve, peripheral nerve, or brain. Okay.
I was just curious about, in some cases, in myotonic congenital dystrophy, you can have polydraminus, and like a clue to think about it. In the whole cohort of patients, polydraminus is common, or it was only common in certain diseases?
You mean the question is about polyhydramnios or DMPK?
DMPK, like if it was only associated with DMPK, or other genes could have also polyhydramnios.
So in my knowledge, for polyhydramnios, the only case I know, but maybe Joanna, is DMPK.
So it could be helpful.
But I think it's important to, in this case, whatever the case, to examine the mother and the parents, and to perform the test, because the whole exome is
not pick up.
Not sufficient.
And sometimes when you do PCR in the baby, it's normal because the expansion is so large that you can only see one.
This is diagnostic lab. You have to, yes, to mention that you have to target.
Thank you.
Do we have any more questions from the audience? This seems not to be the case, so we thank the presenters once more. And wish you all a nice evening. Enjoy the evening.